Good morning, Dr. Chong. My name is Xian Ding and my metric number is 79538. The poster I am presenting is about exploring the potential of the F5512, a herbal extract for hair growth and hair health enhancement. Alopecia is a common hair loss condition that affects both men and women. Current treatments such as minoxidil and finasteride have side effects. Therefore, researchers propose that herbal remedies is a potential alternative treatment option for alopecia. The F5512, a formulation of 6 herbal extracts from traditional oriental plant, as shown on the left side of the poster, has potential benefit for hair growth. However, there are no report of its effect on human hair growth. The objective of this research is to investigate the pharmacological effect of the F5512 on the proliferation of human dermal follicular cells in vitro. For the methodology, first is the isolation and culture of human dermal follicular cells. Human occipital skin specimens were obtained from hair transplantation surgery. Samples were dissected into single hair follicles, and human dermal follicular cells were obtained from individually isolated hair follicles. Human dermal follicular cells were transferred into petri dish and cultured into double-course modified agar medium, supplemented with penicillin, streptomycin, and fetal bovine serum in a humidified incubator set to 37 degrees Celsius that contained an atmosphere of 5% CO2. Second and third passage of human dermal follicular cells were used. Second is the viability assay. Cell viability was determined using MVT assay. Cells in 100 microliter of grow medium were seated into each well of a 96 well plate, allowed to adhere for 24 hours, and then treated with serial doses of the F5512, which ran from 0 to 10,000 ppm, or 1 micromolar MXD, which served as the positive control, respectively. After the treatment, the media was removed and replaced by phosphate buffer saline containing 5 mg per ml MDT. The plate was incubated at 37 degrees Celsius for 4 hours and the remaining supernatant was removed. 100 microliter of DMSO was added to each well and mixed roughly to dissolve the crystalline formation. To ensure all the formation crystals were completely dissolved after 10 minutes incubation, the optical density at 540 nanometer was determined using an ELISA reader. Cell viability was calculated as the mean test adsorbent divided by the mean control adsorbent multiplied by 100. Last is the proliferation assay. Ki67 expression was determined as a marker of cell proliferation. Human dermal follicular cells were incubated on the cover slip in the presence of 100 ppm DF5512 or 1 micromolar MXD for 24 hours. The cells were fixed, immobilized, and blocked. The cells were incubated with an anti-Ki67 antibody overnight at 4 degrees Celsius, followed by aloxafor 594 conjugated ghost anti-rabbit immunoglobulin G for 1 hour at room temperature. The human dermal follicular cells were then washed and counter stained with 1 microgram per ml DAPR for 5 minutes and mounted onto a glass slide. Images of fluorescence were acquired using confocal laser scanning microscope. For the result, the graph on the right side shows the viability of human dermal follicular cells after 24-hour and 48-hour treatment with various concentration of the F5512. The F5512 at a concentration of 100 ppm enhanced the viability of human dermal follicular cells compared to 10,000 ppm of the F5512. The F5512 increased the level of Ki67 in human dermal follicular cells, thus promoting the proliferation of human dermal follicular cells. This is because Ki67 is an indicator of the proliferation of emergent hair follicles. At the bottom are the reference I used in this research. And that's all from me. Thank you.